Hello, my name is Tobin, and my presentation is called A Ton of Information About Anhydrobiosis in Bdelloid Rotifers. We're going to start with a refresher on what a rotifer is and where they live. Then I'm going to introduce this really cool concept and ability that rotifers have called anhydrobiosis. Then we're going to break that down into a few key parts like trehalose, which is this really important sugar in anhydrobiosis, and this really neat DNA repair mechanism that is responsible for all these other cool aspects of rotifers. So their resurrection, quote unquote, from anhydrobiosis, their ability to survive ionizing radiation, and the reason why they're fully asexual, at least Bdelloid rotifers are. And also tied all together with some horizontal gene transfer of why they can do this all, where they got the genes. So a little recap of what rotifers are. They're myofaunal, so like really tiny. And they have a corona for feeding. They've got their body covered in a cuticle, and they've got a single foot on the back. Bdelloid rotifers are the ones I'm going to be talking about for the rest of this presentation. They're a monophyletic clade of obligate parthenogenic rotifers, meaning that they all reproduce asexually completely. Their sister group is Monogononta. All bdelloid rotifers live in freshwater environments, and these are typically limnoterrestrial ephemeral habitats. So that is just like places that should be damp usually, like on moss or in wet leaves, but those places can often dry out. But what happens when their habitats dry out? They are aquatic. They need water, right? Well, that's the fun part. Rotifers can do what very few other animals can. They can survive desiccation. So some or other organisms like mosses, some fungi, some larval stages of animals like the statoblasts of ectoprocs, they can also do anhydrobiosis. But only 3 groups of animals can do anhydrobiosis at any stage in their life. And those are bdelloid rotifers, nematodes, and tardigrades. Anhydrobiosis is the process of surviving through extreme water loss. A rotifer that is subject to extreme conditions, which is usually just like when that layer of water on the moss evaporates, it'll undergo anhydrobiosis, curling up into a little ball that is called a tun, hence the name of this presentation. This is basically the smallest the rotifer can get while still fitting in all its organs. And shrinking up like this lets them regulate the rate at which they lose water so they can have a bit more time to get into their tun state. And what's really cool is their metabolism drops by like 99.99%, and I'm not even kidding, like it drops to basically nothing. And that is really helpful for them to be able to survive a really long time and essentially not do anything except exist. So typically, the harsh conditions are just drying out for like a day or something. But this year, a paper was published that talked about these rotifers that scientists thawed out of some ice that were perfectly happy to keep living once they were thawed out. But the catch is, that ice and the rotifers in it have been frozen for 24,000 years. Most anhydrobiosis in animals uses this non-reducing sugar called trehalose, or a sugar that doesn't really react with much. And for a long time it was thought that anhydrobiosis and trehalose go hand in hand. But until 2015, and with much effort, no one had found any trace of trehalose in bdelloid rotifers. But when they finally did find trehalose, it was in much lower quantity than they were expecting to find, and the genes were, well, really weird. Like, we'll talk more about that soon, but the quantity of trehalose was measured to increase as the rotifer entered tun state, which is helpful to know, but it's still lower than normal, indicating that it's used more likely to be a signaling molecule and not so much as cellular integrity as it's often used in other animals. Desiccation results in many breaks in the DNA, which is really not ideal in most cases, but this has led to a really strong selective pressure for rotifers to be able to put their DNA back together. This lets them be resurrected from being in tun state after a bunch of their DNA has been broken by that drying out. A dry rotifer, that is, a rotifer in tun state, is basically no different than any other speck of dust, like physically speaking. So if a breeze comes along, sweeps up that dust, the rotifer is going to be going along with it. that is really great for dispersal, but it means that the rotifer who's suspended in the air with the rest of the dust is going to be exposed to a lot of sunlight. And sunlight has a lot of ionizing radiation, which is famous for messing up DNA and breaking it apart, which is why we wear sunscreen. But a rotifer cannot wear sunscreen. It can't really produce many melanin or many pigments either. So it's going to use that same repair mechanism instead of a preventative mechanism. So it repairs its DNA after the ionizing radiation has broken its DNA apart. And this means that it can survive absurdly high levels of radiation that most things can't. Typically, genes are passed from parent to offspring, right? So you'd think that where Bdelloid rotifers got their ability to survive desiccation would be from that common ancestor of all Bdelloid rotifers, because it's a monophyletic clade. But Bdelloid rotifers got their genes for surviving desiccation from plants, which is kind of weird. So these genes, getting these genes from another organism is called horizontal gene transfer. And that's just basically when genes are passed from one organism to another that's not in this parent-offspring relationship. So how does this connect back to the DNA breakage and repairage thing that we're just talking about? While the DNA is breaking apart, the cell is also pretty vulnerable to letting in foreign bits of DNA into the cell. So this diagram is an extreme exaggeration of what happens, but basically the rotifer's DNA breaks apart while the rotifer is in ton state, and then foreign DNA is able to enter, and when the repair mechanism is putting the DNA back together, it'll incorporate some of the foreign DNA as well into its chromosomes. This really seems like it should be selected against, but like 8% of rotifers' expressed genes are from foreign origin, including many of the genes responsible for both entering and recovering from anhydrobiosis. These genes include some of the ones for producing trehalose, the enzyme for breaking down trehalose, and some of the genes responsible for that amazing DNA repair mechanism. Another wildly interesting aspect of this rotifer ability to put its DNA back together is the fact that it doesn't put it back together the same way every time. So say this is your chromosome, and typically in most animals that reproduce with meiosis, they're going to have homologous chromosomes, so two chromosomes that are basically the same have the genes on the same spot on both chromosomes. But if it's a Bdelloid rotifer and it goes through desiccation, its DNA breaks apart, and then it puts it back together, maybe the allele or the location of a gene that was once here might accidentally get put back over here. So your homologous chromosomes are no longer going to be homologous because it's still either here or maybe it moves somewhere random else. So this means that their homologous chromosomes can't line up, and meiosis doesn't work because the homologous chromosomes won't line up. This means that just sex isn't possible, yet the species of Bdelloid rotifers are still able to speciate. This is largely in part due to the accumulation of new genes from horizontal gene transfer. And this has allowed rotifers and Bdelloid rotifers to survive without sex for 40 million years. So, in conclusion, we had a recap of what rotifers are, who the Bdelloid rotifers are and where they live. We learned about anhydrobiosis and ton state. We learned about this absolutely incredible DNA repair mechanism, and we learned how they use this repair mechanism to rebound from desiccation and to resist extreme levels of ionizing radiation and receive like 8% of their active genes from non-animal origin, and how it eventually results in their asexuality. Thanks for watching. If you have any questions, please let me know. I learned a bunch of stuff that doesn't quite fit into this presentation, and I would love to share. Thanks for watching.